So David Juncker from McGill is going to talk about microchips and scalable technologies. That sounds like my cat.
Okay, well, thank you very much for having me here. And thanks, Larry, for taking the risk for inviting a physicist engineer to this wonderful meeting. So I'm trying to fit in here and talking about microchips, and I think we had a wonderful talk where we could see basically how the advancement of technologies could really transform how we do diagnostics on this work. And so for all of you working in biology, all the physicists and engineers are doing all the hard work, and then you get to get all the benefits. And so I'll show you some of the hard work we are doing here, and hopefully give you some interest in that too. Okay, and just to show you a little bit what we are doing, so my lab is, as I said, I'm a physicist engineer, working to biomedical engineering. So we'll talk to you about the antibody microarrays, but we do also patterning on surfaces for cell guidance. We work for like global health applications using thread as diagnostics, or for chips for doing also infectious diseases. So if you have interest about this, I'd be happy to talk about it after my presentation. And for the talk today, I'll speak to you about scaling. So I did actually my Ph.D. within a company, which was IBM. And so we spoke a lot about computers and how they were improving power and how there was this scaling law of Moore's law, as I will talk to you about more in details. And what I have really seen is how this has impacted now also biochips. And so biochips, and that's what we're being used, of course, for diagnosing various diseases in different modalities. And the one disease we're working with is cancer. So we just heard a lot about cancer and how their new technologies can improve the diagnosis. And what I will be talking about is antibody methods, in particular, for diagnosing cancer. And there is a big interest in proteins. So proteins are the most used biomarkers nowadays in the clinic. And so there's a big interest in also improving the technologies there to measure them. And I'll show you some very preliminary results and some of the studies we're doing, and then concluding and then giving you an outlook on how the technology could evolve further. And so early diagnosis of cancer is, of course, very critical, and I just learned basically that this meeting was dedicated to Emma Swanson, who has died from cancer because of a lack of diagnosis. And as we know from this, well, as we all know, early diagnosis basically helps survival, and this is kind of outlined in this graph here, where you have different stages of cancer. So if you catch it at a very early stage, your survival chance is actually really high, statistically speaking. And if your cancer is metastatic and dispersed in the body, well, then your 5-year survival rate is very low. So that's the statistics. And of course, we think that by having a better technologies, we should be able to diagnose cancer much earlier and then help the survival. And just to give you a bit of recap, because I'm going to talk about big numbers, and I think this was a general audience meeting, so I wanted to have this one slide to put things a bit into perspective. So we have our DNA, we have 3 billion base pairs, we have 20,000 genes, and these genes are basically then transcribed into RNA and which are translated into proteins. So we have 20,000 proteins, several hundred thousands of variants of proteins, or if you count antibodies, maybe millions. And this is still not completely clear. And then these becomes organelles, and they become cells. So that's the building block of the human body, and which is then assembled in different tissues, organs, and organisms. And cancer and all these diseases are actually diseases that occur here, and most diagnoses are unfortunately only done once the disease is manifests itself or is done at the level when actually it affects really large-scale organs. And beyond that, that's kind of the evolutionary story, but we have even gone further, and this is coming back to chips. Nowadays, we have even an enhanced functionality so that when we want to have a bit more information or we don't remember, we can just hook up on the internet, and this gives you extra power. And this is, of course, one of the outcomes of the scaling of computer chips. So this wonderful, well, this incredible scaling of a technology that started here with integrated silicon chips about in the 70s, and you have this law, so where every 18 months the number of transistors on a chip doubles, and so you have here a linear scale, which is basically the years, and here an exponential scale, and then you have a linear line connecting these two things here. And this is generally known as Moore's law. I think, sure, most of you know about this. And so Moore basically, he observed, it became a law after, because he observed that in the early days that computer chips were doubling all the time, and then the semiconductor industry took this this as a target. So they want to double now the computer chips every 18 months. And so to some extent, it's a bit of self-fulfilling prophecy, except, of course, that maintaining that over such a long period, that's really a fantastic achievement. And that's what has transformed many of the technologies that we use every day. And so one of the technologies, so these were computers 1944. I was just talking with a gentleman here who told me that he had been developing some of the fastest computer back in the 50s. And so this one was really slow, 15.3 seconds for one division, 72 numbers with 23 digits. Well, now we're laughing because, yes, now we have cell phones. I just came from Canada. I have to do some routing here. And I can show you one, too, if you're interested. And so we can, and these are actually small computers which can do a lot of things for us. But if we look back now, took a broader perspective, if you look at biological systems themselves, they're actually really, really complex too. And there's also scaling going on here, because we all start as a single cell. And that's really one of the most fascinating things to me. A single cell, like Gordon Moore here, became actually 10 to the 14 cells, which is a pretty large number, and it has about 10 to the 11 neurons, which give us some sort of intelligence sometimes. But so that's kind of, well, that's the complexity of the system we're looking at. And so, we can compare to the numbers of stars in the Milky Way or the world population, and we see actually that the human body itself is something of extraordinary complexity. And so we have this great complexity, and I showed you this before, this machine here, which was the early computers. And so when this great complex system like us goes sick, and if you go to the hospital and they take some blood, the type of systems that we'll use to analyze whether you're sick or not is kind of this type of machine here. From performance-wise, it lines up a little bit with this one, actually. This is a few years old, but we can see that, well, this is kind of still the prehistoric age of testing. And of course, there have been big progresses, and especially in the research, but also already in the clinic, as we heard just before. We have now chips, and these chips are the foundation now for making integrated systems that can do much faster testing. And so we had now the scaling of computer chips, and so I want to just superpose you now the scaling of these DNA chips. And so DNA chips, they were actually proposed back in 1994 by Pat Brown, and they started with a chip with 400, and then they had a dramatic progression up to 6.4 million chips here, or probes on a single chip, which can then be used for, well, research, first of all, and more recently also for disease treatment and prognosis. And this is now, so for example, in Canada, in Ontario, if you have breast cancer, so many women have a benign form of breast cancer, but because they didn't really know, they would give them actually aggressive therapy, and which had, of course, morbidity, so really negative side effects. But now using, in this case, they have a multivariate panel chip with 17 gene expressions, which measures the expression of 17 genes. So now they can basically measure whether the woman will benefit or not from this therapy, and if they don't benefit, then they don't give it to them. So in the end, this is actually a case where a test also really helps save costs in the healthcare system, which really was what helped the rapid adoption of this test in Ontario in this case. Now, the other technology that is coming up is genome sequencing, and so we had just a discussion about this in the previous presentation. So the first time the human genome was sequenced started in 1988, and it's a project that lasted for 13 years. It took $1 billion. It took thousands of scientists, and this is a technology that has progressed in an amazing way, where nowadays on a one chip we can sequence up to 600 gigabases, so that's about 200 times more than the bases we have in our body, and the cost. So if you want to sequence yourself, and I heard that from a colleague who's working at MD Anderson Center, so the patients who come in, the wealthy patients, they actually send their sample out to have them sequenced, and then they come in with their sequence, and they ask the doctor, can you do something with my sequence nowadays? And yeah, so we have the information, we don't really know how to act on it, but we saw in the previous presentation that this is also something that's coming. And so this technology here, and has actually have a progression that has even been more dramatic than any of the other technologies. So a rapid rise when these so-called next-generation sequencing methods became available and have had really strong transformative effects. But what we are made of, what actually drives us in everyday lives are proteins. And yet, so proteins are shown here. So this is kind of a schematic of a cell with the flagella and the gooey things inside. And so these proteins are doing all the work inside of your body. So we have catalysis, which is done by enzymes. We have signaling with receptors. We just saw that before. We have structural support. We have regulation, binding to DNA. We have immune system antibodies. And we have also mechanical work, like outlined by this flagellum. And so obviously, when you have disease, actually what's in your DNA often gets amplified at the protein level. So if you had a good technology to measure these proteins, that could be, of course, very interesting. And also, to kind of argue in this sense, so we have, as I said, 20,000 proteins overall. Actually, most of the drug targets nowadays go for proteins or towards proteins, yet we're only targeting a very small fraction of the proteome, and there's still many questions that remain about proteins. But as I said before, proteins are also yet the most widely used biomarkers in the clinic. And they're, of course, based on clinical tests. So just a little recap for biomarkers, because biomarkers actually can be many different things. And so there can be biomarkers for risk of disease. There can be biomarkers for early diagnosis of disease. And then once you have disease onset, you want to have disease stratification, so prognosis. So is it an aggressive or is it a benign disease, which, of course, will affect your therapeutic regimen, and which can then be matched. So we can also try to predict whether actually this will be really efficient. And then you'd like to measure therapeutic efficacy, as well as ultimately, once you have been, if you have been treated, whether you might have a relapse. And so some of these are not time sensitive. Risk for disease, you can do that at one point, so it's typically mutations. But many of these, like, will you be developing disease, with something that should be measured continuously, because you don't know, of course, when it will happen. And so for this, and we saw that in the previous talk, what you'd like to measure is blood, because the blood, it's both what supplies all your organs with nutrients, and it's essentially also the waste of your body. So if you have some organ that has some dysfunctionality, and if it excretes this will likely be found in the blood. Now, the great advantage of blood is it can be easily sampled. I mean, it's not entirely non-invasive, but it's mildly invasive, so it's something that's also very well broadly accepted, and so that we can have this sample repeatedly and many times. And so we have then some methods that have also developed to identify proteins, also not directly in the blood, but I'm going just to make a link here. And the one technique which will not talk more about is mass spectrometry, which can measure large numbers of proteins and has done a lot of progress. And the other technology, and here we're coming back to chips, are antibody microarrays. And so antibody microarrays, you would immobilize the probes on the surface, incubate your sample, and then each antibody here would bind specifically to one of these targets, and then you can measure whether something bound on each of the probes, and then identify whether that target or that analyte was present in your sample. And if we look now at the progression of these technologies, mass spectrometry, it's not here, but antibody arrays, so they started, well, a bit earlier than here. I think this is just late slides, but so they started here, and we have now arrays that go up to about 1,000. And with mass spectrometry, same thing. We can measure actually several thousand proteins at once in a sample. And so following these efforts, people have now been able to actually look at different samples and try to identify potential biomarkers for all these diseases in these different samples. And out of the 20,000 proteins, up to 5,000 have actually been identified as candidate biomarkers for various diseases. But out of these, there's a huge bottleneck here. Only about 109 have been approved as a biomarker use by the FDA. And if we look at the approval rate, so since 1999, we have these thousands of candidates. The approval rate of biomarkers for different tests is shown here. So that's from a paper by Anderson. And so we have the year of approval and the number of biomarkers that are being approved each year. And so despite technological advances in identification of biomarkers, we haven't really improved on the rate of biomarker approval. So in 2006, there were no biomarkers approved. In 2007, there were 3, and here there were 2, and okay, the following years, I don't have the numbers. So there's basically one problem that still hasn't been resolved out of this data here. And the challenge is, of course, also that when we look at blood and want to measure biomarkers, as we discussed before, blood is really your waste, so it's kind of finding a biomarker is finding a needle in the haystack, to put it in a nice way. And to put it in perspective, so we have a concentration of blood here, which is 10 molecules that are 10 to the 11 or 10 to the 10 roughly, and in picograms per milliliter, so we have 10 to the 10 picogram per milliliter for albumin, for example, and the molecules at the low end here, so which are interleukins and so on, they are 10 to the 0 picogram per milliliter. So we have a dynamic range in this case of 10 orders of magnitude. And so to relate it, the human population is coming close to 10 billion. So identifying a biomarkers inside of the blood is like identifying one person among the entire world population. But of course, for a test, you want to do that in 30 minutes. And actually, that's something that's possible using a certain test. But to come back to our biomarker identification technique, so we have this shotgun mass spectrometry, which can only detect molecules at this stage here, or we have these antibody arrays, which can detect them here down to this level. So of course, when you want to measure them after in blood, it's very hard to find them. But there's these sandwich immunoassays, which actually have the possibility of measuring molecules at a much lower concentration. And so I'm going to be a little bit technical here. And so despite being actually with a physics engineering background, if you want to do some progress in some areas, you actually have to really understand how things work. And so here I'm going to explain to the one who don't know how an ELISA works, and to the ones who know, then it's interesting to listen to it so that you actually can relate to the next part of the presentation. So this sandwich assay, that's actually for most clinical tests, that's the one that's being used. So they have a probe that's immobilized on a surface, a capture antibody, then it's rinsed, they would block the surface here with some agent. Then there would be a sample incubation step, so that's usually when you buy a kit, that's the step where you start with, so you apply your sample, it incubates, and then your antibody will specifically bind your targets, which are outlined in red. But because, as we just learned, we have some proteins that are 1 million times more concentrated, or 10 million times more concentrated, even if it binds very weakly here, this might still bind on the surface, or it might just stick on the surface here. And so if you just measure at this point what you were binding, like in the single antibody assay, you would have 2 false positive signals here, which basically would skew your assay results. And so what people then came up with is with a sandwich assay where they introduce a second binder, which now again binds the same molecule, but then from another part of the molecule. And in this way, they can discriminate, because it's very unlikely that this one will also bind non-specifically to the few other cross-reacting molecules. And in this, you have then a binding of these second molecules, these detection antibodies, and they can be labeled with some label for a readout. And so in the end, you measure a signal which is proportional to the concentration of your targets inside of your sample. And so, well, that's a great format, so why don't we do just an array with this format? Now, that's exactly what people did. So they took like this classical ELISA, where you immobilize an antibody, you apply your mixture, you apply a detection antibody, and they did an array format. So now you make an array of little spots on the surface, you apply your sample, but now here, because you have many different spots, you have to apply many different detection antibodies to bind onto these spots. And so this type of technology actually started 1988 here, just a bit before the DNA microarrays. And this is a logarithmic scale, so in the span of about these 20 years here, we have gone from about 4 targets to 40. So it's a bit less impressive than sequencing. And so why is that? Why can't we go further? And so I'll show you some examples of why this is happening. So one is actually clinical study. So they used 120 proteins. I mean, they made different wells, so they didn't have too much mixing. It's a commercial experiment they did, and they were looking at these biomarkers for Alzheimer here in plasma sample. And so this was an important study in 2007, but there was a News and Views, and I think a follow-up article showing that this data was actually reproducible. So these assays didn't work, and so the author was arguing that the antibodies were not reliable, and I'll come back to that later. And ultimately, what I think is the reason here is that there's a fundamental flaw in the way the assay was designed. I'm going to try to convince you that. And so, something that works well at a small scale, when you have an ELISA, 1, 2, 3 works well, but when you want to scale it up, well, sometimes it doesn't work that well. And so to give a bit of counterpoint to the Rube Goldberg rule, Albert Einstein said everything should be made as simple as possible, but not simpler. And so that's kind of a case where people wanted to go straight to the goal, just made these simple arrays and threw everything on it. And well, that's maybe a bit simpler than what you should be doing. And so the reason now why this is not working, that's what we have been working on for the last few years, trying to understand that. And people kind of know that these assays are very difficult and they have cross-reactivity. So what we try to do is just to kind of rationalize and enumerate it. And the way to do that is that, well, so just to explain you, so here is kind of an example of 2 spots. It's a sandwich assay. And so what we're looking at is how these antibodies from this spot can start to cross-react on this spot and then basically create false positive signals. And look at all the combinations that exist. So if you have an array here now with n targets, let me step back here. So, the first scenario of cross-reactivity is that the detection antibody here can cross-react with this antigen that is bound here. So that's just one thing that can happen, and that actually happens. So now if I have an array with n targets, so I have n minus 1, so you have 10, if I have an array with 10 targets, I have 9 other detection antibodies which are applied on this assay and which can cross-react. And because I have n spots, so I have 10 spots, this can happen on each one of these spots. So for this scenario, you have n times n minus 1 combinations, or liability pairs, that's how I like to call them, which basically can give rise to a false positive signal. Then another scenario that I have is that actually the detection antibody can directly bind to the capture antibody here. And here, same thing, I have n times n minus 1 combinations. Now another scenario is that, because I'm always mixing all these reagents, is that these detection antibodies can actually bind between one another. And here, it's combinations among the same group, so there's a factor 2. Another scenario is that the capture antibody can bind to the antigen. So here, it's this one, it cross-reacts to this antigen here. And again, this gives you a false positive signal. And finally, the proteins, they like to make complexes. So if you have a complex, you again might generate false positive signals. And so if we add this up, that's 4n times n minus 1 scenarios of liability pairs. So each of these combinations is a liability pair. And so if you have an array of 14, that's 728 combinations. If you have an array of 100, that's 40,000 combinations. And so if one of these combinations creates a cross, so if these molecules cross-react, then that's a false positive binding. But that's just theory. And so we wanted then to see if this happens. And to some extent, we already know. So this is now these graphs where we test this cross-reactivity. I'm going to skip a few. So these are 14 arrays with 14 targets, and we just test each of the detection antibodies here in a complete negative control. And if all was fine, this should be just a white, nice board. But here, when we get cross-reactivity more than 20%, it becomes red. And so we do this test without antigen, with antigen, and when we apply a mixture of detection antibodies to a single antigen. And as you can see, we have a lot of cross-reactivities. And so there's companies making these kits, and they spend their life trying to optimize that, to minimize it. I mean, not all of this is a false positive signal. Some of it is just background noise, but this has a huge effect on your assay. And ultimately, your sample is unique. Each sample is unique, and each sample might give rise to its own cross-reactivity. So overall, this is a very vulnerable. So if you think about engineering, this is a really vulnerable assay design, which is not robust in an engineering sense. And so that's why basically many of these assays don't work, but and why they can't be scaled up, because this is with 14. Of course, if you do this with 100, then this would just become all red. And also, if just one antibody is actually behaving badly, your entire array is basically compromised. And so what we kind of did then, and so here come back the microfluidics liquid handling side, is that, well, we started like here. We apply the sample, but then we deliver each detection antibody individually on each of these spots. So kind of conceptually very simple, practically a little bit more difficult, as we'll just see. And so the way it works now, the process is we start with the array, we incubate the blocking solution, we incubate the sample, and then instead of incubating this mixture of detection antibody, we have to come back and spot and align on each of these little spots so that we can apply the detection antibody. So the great advantage, we have no more combinatorial interactions, so all these scenarios I show you, they are gone. We even save on reagents. We only use 1 nanometer of these antibodies, and well, the sample is still a bit bigger. The challenge, we need to align, so we need to spot on the same spot, and well, that people don't do that normally, so you needed to develop that. Also, when we spot such small volumes, is our assay actually still working? So that's the type of things we have to work on. And so I'm just showing you a little bit. So this is kind of a custom-built spotter with a special alignment feature. We make pins out of silicon here. And this is an example of the overlay spotting where you have the green and the red. And you can see the overlay here where we have a nice alignment. And so most of our life is now spent on optimizing this. And I'm not going to show you that. And that I'm thinking you'll be thankful for that too. But what we have been doing this so far is scaling this platform up to 50 targets, and I'll like to go further, and I'll show you the challenges for doing that. And with these 50 targets, we can then start to measure proteins in samples. And so here's an outline of such a slide. So here's a slide, and we have actually 16 wells. On one slide, we can do 16 experiments. And in each of these experiments, we can measure different samples or make a dilution series to have a calibration for our assays. And so we then did a first preliminary clinical study with a colleague looking at breast cancer samples, which were a bit more advanced cancer on one particular subtype, and trying to see if we could see different protein profiles, kind of a different signature of proteins in the blood of these cancer patients with the controls. And out of our preliminary measurements, we saw some differences. And when we did what's called clustering analysis, so grouping the different samples based on the concentration profiles on their signature, we found that basically the controls and the cancer patient sample, which are outlined with this blue line here, were really nicely separated in two cohorts. And so that's kind of our first study. And so we're having other studies going on, and one of them, for example, is also traumatic brain injury, which in Canada is more largely focused on hockey, and here I think it's football. And so here we actually do time course studies where we take both the plasma, serum, CSF, and because we work with a surgeon, they're actually putting a microdialysis probe inside of the brain so we can get access to that too, and then measure the response of these molecules over time. And the interest is that these patients come in, and right now there's really no diagnostic or understanding what drives the outcome. And so we hope that by looking at time course, we can recognize some patterns that might then tell us whether there's a good or bad outcome. Now, one of the challenges is that our ACM is kind of a big platform. It's not really very convenient to use. And so one of our efforts, technology-wise, has to make this much more convenient that some of you might adopt it too and get then some of the glory I was talking about. And the way to do that is now that we don't, so now we have to spot during our experiment. So what we're doing here, it's actually quite simple. We just spot both the capture and the detection antibody. Then we can store them in our refrigerator, or we're working also on, well, room temperature storage. Then when we have our sample, we would take the chip out. We have these two chips. We transfer them in a way that colocalize the reaction, the reagents here, so that there is no mixing. And then there's a binding that can happen, and then we would read it out in a classical way. And this is then the little apparatus. So this is version 1.0. This is version 2.0. And using that, we can then make these nice assays. And we are using this now for doing time course studies of cancer development in mice models, basically. And so what's next? So this is kind of current status. One of the things we would like to develop is improve our platform. So here I'm really speaking about the technological developments. And one big opportunity we're seeing is to measure post-translational modifications. So we know that in many diseases, so the way the proteins are processed, so they can be added with glycosylated or phosphorylated, or they can be cleaved. And that's actually a very strong indication of disease. And with our platform, what we can have is the same capture antibody and have multiple replicate spots, and then apply different detection antibodies, which can then start to probe the nature of the protein that is bound on the surface. And I think that's actually one point where our platform has a big potential, because that's something that could not be done easily on a chip right now. Now, the other story, and that's something we're also working on a little bit, is that I showed you that the dynamic range is 10 to the 10, and that's actually not true. I told you something wrong, but that's what actually the community says, because that's kind of a value that's given. It's not the value that's in blood, it's the value that we can measure with this type of technology. But if we step back a bit and we just kind of do a back-of-the-envelope calculation, we have 3 liters of blood, roughly 1 liter of plasma. The highest concentration of molecule is 10 millimolar, roughly. The lowest concentration is basically 1 molecule in 3 liters of blood. That's the limit that we can think of. And that's, for the ones who know, that's 1 yoctomole, so it's 10 to the minus 24. I had to look that one up. And so if we look at the dynamic range of proteins, it's actually 10 to the 21. And so we can measure now up to here, down to here, but if we think back, if you have a small tumor, you want to detect the tumor early. And so in breast cancer, for example, tumors are detected by mammography at 5 millimeter in size. If you calculate how much, well, you can calculate what's the excretion rate of these tumors in the blood and how much proteins you would find in the blood. Because if it's a good biomarker, then that's not something that's there before you have it. It's something which would be really specific to the proteins. So if you do that calculation, then you get a bit depressed, because it's really low concentrations, and it's much lower than the concentrations that would be here. So So I think the biomarker that we have actually sampled so far are the ones that are in the high range, but that's probably not the best one. So the best biomarkers would be probably much lower, especially if you want to do early diagnosis before you have a huge tumor. You will have to have technologies to measure much more down. And there is a technological push also going to that direction. So people are developing immunoassays where they can detect single binding events. And by doing that and having really good antibodies, they can actually push this down to femto attomolar concentration. And so that's what I think. If we are able to look there, we have a much better chance for finding actually much better biomarkers. And so there's still big space to be explored. So it's a bit like having a better telescope. If you have a better telescope, the universe becomes bigger. Well, here we kind of know the size of the universe, but I'm pretty convinced that if we look deeper, and I'd be willing to bet something on that, we will see new things. And so this brings me basically to the conclusion. So the one point I'd like maybe a go-home message to is one of scalable technologies. So I showed you different types of scalable technologies. Now what's great with scalable technologies is that once you've had it, you can just make incremental progress. And if you do that incremental progress, like making it twice as dense, and then again twice as dense, and again twice as dense, and by doing that a few times, you can actually create something that's completely transformative and disruptive. And we have seen that with computer chips. We're seeing that with DNA microarrays sequencing. And if we can replicate that with protein chips, I'm sure this will have a very strong impact too. And so that's why one strong motivation for working on our scalable protein platform, where we can then quantify hundreds and hopefully in the future thousands of proteins in small volumes with very high limits of sensitivity. And this will then really lead the way to make new discoveries into biomarkers and then help them at the different stages of disease management. And so the outlook basically is then antibody reagents. And for all the ones who work with antibodies, everyone complains about antibodies. But if you have a great antibodies, then that's worth gold. And I think there's someone here who knows that, who's in the audience, who knows that very well, that if you have good binders, they can have actually a huge value, both commercial, of course, both in terms of research. So I said we have 20,000 proteins. There's only about 5,000 antibodies that are available against these. There is maybe 1,000 pairs, so we need the pairs that are available, so that's a limiting factor. I mean, if we're able to acquire them in the first place, because when you want to acquire 1,000, it's a lot of money. But so we'll still be limited here. But there is actually a possibility of making a lot of antibodies, and there has been this effort by Matthias Julen, which was, I think, speaking here last year, which now has produced antibodies against 15,000 proteins in 10 years for a cost of about 100 million. So it's a bit similar to the Human Genome Project. I think where there is a big challenge for budding scientists is can we transform the antibody identification and production using new technologies so that we could produce many of these binders in a particular way. And I think Larry had the vision for this, and he has actually developed technologies to create binders in a large scale and also helped in identifying biomarkers against some of the diseases and many others that I haven't mentioned here. And so if we have these binders, then I hope that we can scale this up. And I said, well, single sandwich assays or sandwich assays are here at the rule of 10. If we get these antibodies, I think the scaling could happen quickly, and the quicker it is, the more impactful it would be. Of course, then we will see how this will play out. And with that, I want to thank all the funding agencies, and yes, my group kicks ass. And I want to thank you all for your attention, and be happy to answer any question.
So over the decades, there have been a lot of Moore's Law skeptics. And in fact, we still get every so often somebody says, oh, it's over now. And the reason we mostly believe Moore's Law will continue is because it's been going on for so long. Not so in the sequencing and in these other worlds. Most of these rapid advances have been very quick. I know people who run genome centers, and they are all moaning to me about how Illumina is now raising prices. And so I want to know whether you really, in your heart, believe that those quick blips of exponential growth are really sustainable.
Right. So, I think, so what I've seen in computer chips, it has been astounding how long they have been going. Now, their rate has been slower than what we have seen in DNA microarrays. And actually, in DNA microarrays, they have hit the wall. I mean, maybe I didn't mention explicitly, but they went up and then they stagnated. And the stagnation there, I think, was twofold. On one hand, maybe there was no demand for higher chips, or they were too expensive. And of course, sequencing came along, and people were just switching to sequencing. And with sequencing, yeah, we also see some limits there in terms of scaling up. But there the challenge, I mean, is the need for really scaling up more, or is the need elsewhere? So that's all. And I think there's still room, on the other hand, for actually reducing the cost in the long term to make some technologies that are then more widely usable. Yes.
Back up here, yeah. So it's really loaded with potential. It's very exciting for personalization, and also it could easily incorporate something like somomers in the printing. So you're not limited to the typical antibody problems.
Right. No, I haven't mentioned, but I said detection antibody or capture antibody. In principle, you could mix and match different binder modalities to expand your repertoire, basically.
So I'm just wondering, what kind of controls do you incorporate routinely?
Oh, so there's no routine here yet. There's mostly development at this point. So the challenge is for us, so as I said, most of the work is in optimization, our assay conditions and so on. And at this point, the antibodies we have used are commercial antibodies, which are used in sandwich ELISAs. And so we primarily have been relying on the vendors' quality control. But we're also pushing into the direction of doing like imaging SPR type of methods so that we can then get a better handle on our antibodies. Because that's if we want to have more antibodies, we need to be better able to actually qualify and characterize them. Because that's also one of the bottlenecks at this point.
So you talked about a little bit with the glycosylation between aberrant and normal glycosylation. So there are many, many protein modifications. And I'm just wondering what your thoughts are on picking up those modifications in like a large-scale screening type situation.
Okay, that's a good question, and I'm not sure I'm the best person to answer it. Okay, in terms of glycosylation, what's the big challenge is actually to have specific binders for these different glycosylations. Now there is different binders, they're not very specific, so you could scale it up. I mean, we have the possibility of scaling it up, and then by scaling it up, even if you don't have unique binder specificity on each of the individual binders, you might still be able to see some sort of patterns that come out by having mixtures of binders. Now that's an area we would like to push, but we haven't really developed it very much yet.
I enjoyed your presentation. I'm a great proponent of all the methodologies that we are developing to do really high content patient data capture. And that comes out of a diverse set of terrains, and they're technologically, I would say, advanced. What we're missing at the other end of the spectrum is the optimal mining of this data through harmonized patient databases. And hence, the mileage we get is limited by what we compare it to.
Right.
You speak a little bit to efforts in Canada or elsewhere based on which we can cover that spectrum of our data management.
Yeah, so I think that's a very good point, is how to make some centralized centers or some harmonized centers for data analysis. But this actually goes beyond, for example, for sample collection. I mean, I have to go talk to the clinicians to get their samples, and it's all a bit single shop efforts to even collect the samples at this point. I mean, there's these Norwegian studies that were mentioned where people then have these large-scale studies with large-scale databases where they can both have a harmonized sample collection and then can have also streamlined data analysis. At this point, I mean, we're collaborating with researchers within McGill, but I'm not on the data analysis end, really, so to speak. So I'm trying to provide the technology for doing this analysis later on. But yeah, that's certainly something I would love if the funding agencies could make something that would a more harmonized way for doing that.
Quick question on looking at other technologies. This reminds me, if you go to other conferences that have nothing to do with life science, they have the exact same problem. And the two different ones that I've seen is inkjet printing, and all the folks that are dealing with inkjet printing and micro-manufacturing and MEMS and GPS MEMS chips, identically the same things. curious if you're talking to them. And the other one is all the optical stuff that's happening in photolithography in semiconductor manufacturing. They have all kinds of really clever optical tricks. And when I talk to those guys, they're not ever thinking about bioscience. And nobody's ever talked to them, and they're not talking to anybody. Perhaps a phone call or a visit to some of those guys might help you.
Right. So thanks. So okay, I think there's, so try to answer your first question. So and I think they're both related. So the delivery of the samples onto the surface, so this very small droplets, yeah, that's still a big issue. So there's not, the inkjet spotters you get for delivering biofluids, they are not very high reliability, which is why we went actually to the pin spotting route. And the market for this instrument is quite small, so there's not that many efforts in developing actually better instruments for that. Now the second point, which is a bit related, so is okay, why don't we use photolithography? Well, I think the big challenge is that when people do photolithography, they do multiple levels, and at each level they have one chemical which they apply. Here it's the opposite. We do like one or two, well, 2 spotting rounds, but on each spotting rounds we want to apply 100 or 1,000 different chemicals. And these chemicals are very sensitive, so you can't use the common process that are used in photolithography to process them. But having said that, I'm sure we can, people who do semiconductor chips, they have alignment within 30 nanometer or so, and that's something where there's much room for improving the technology here. So if we want to scale it up and have not 10,000 but 100,000 spot on a slide, then we will have much better alignment. But at this point, actually, what's our challenge is the delivery of the sample, optimization of the assay conditions, obtaining enough reagents. So that's actually our first, that's where we have most of our efforts dedicated at this point.
Okay, thank you very much.
Thank you.